Okay, everyone. So, finals on Monday at 8 a.m. Hope you've all been studying very hard. Been having luck with that. So, I've been studying a lot myself, and I have been able to put out many videos. But let's try and finish up the series. And on a good note, it's a good review for me. I know it'll help some of you. And I'm very happy to always hear this helps. So, HIV. So we covered retrovirus in the previous videos, and I split those into like individual videos. This will just be in the long form, not in the short chop up form. So HIV. HIV is a retrovirus, and the reason why we covered this separately is because it's an important human pathogen, but also because unlike retrovirus, it does not cause cancer. It's actually a lymphotrophic lytic virus. There you go. Meaning that when it affects T cells, it doesn't turn T cells into cancerous T cells, it just kills it. Now, Samuels introduced HIV by starting off with a tangent. So, rule number 1 is introduction. So I'm going to try to output these videos as fast as possible. So I may skip over some details. I'll try not to, but I'll see why I deem as worthy of this video. So, discovery starts out with avian leukemia virus. So avian leukemia virus is a virus that infects birds. And what scientists found out about this is that it does not have an oncogene. However, it was still able to cause cancer. So unlike retrovirus or polyomavirus, it did not cause cancer by having its own gene product that causes cancer. What it simply did was it inserted itself next to a normal cellular growth gene, and it caused that normal cellular growth gene to be overexpressed. And when it got getting overexpressed, the cell became cancerous. So that is the best. So I'm going to write here causes cancer without oncogene. And I'm going to draw a picture here. So I can reserve the right side of the board for a picture. So this is best illustrated with a picture. Here we are. This is a cellular gene, or DNA, cell DNA. There's this cell gene called c-MYC. C is for cellular, MYC is for whatever the heck that stands for. Just know that it's for growth. It's an oncogene, meaning that it tells cells to grow. These kind of genes are heavily regulated because you don't want them becoming cancerous. Now, ALV does. ALV is, this triangle represents the ALV genome. So, ALV genome. And so its insertion here, ALV inserts here, it increases c-MYC expression. And increased c-MYC equals cancer. And this kind of mutagenesis is called insertional mutagenesis. So there are a few ways that you can cause a transformation. You can do it via ligand, via receptor, or a transactivator, or through insertional mutagenesis. So you may see a question like that in the practice final. I'm sure we'll see something like that on the final. So with that in mind, I'll keep the picture there and we'll erase this. So Roman numeral 2 is, I'm going to abbreviate this to make the video go faster, it's human T lymphotrophic virus. Abbreviate HTLV. Human T lymphotrophic virus. Infects T cells. So what's important here is that HTLV was found to infect T cells, and it increased cell proliferation. But, or not but, not 1%, 1% of HTLV patients got cancer. So you can see how the discovery of something like ALV kind of led to the discovery of HTLV, which is why Dr. Samuel started off with ALV. Because he's trying to illustrate, people found ALV and he's found this virus that causes cancer, and it does so by insertional mutagenesis. And they were studying humans or people, and then they saw, oh dang, there's also something that's very similar to, there's a human analog to ALV. So there are multiple types of HTLV. So HTLV3 was discovered next. In case you're wondering why my genes look really worn out, it's because I accidentally used my good genes during research lab when we were cleaning stuff with bleach. And so I threw, that's in the washer. So I have to wear my bad jeans. Just got a bit sidetracked. Anyways, there's just a giant hole right here. Whatever, anyways. So continuing on with what we're going to see, they found that HTLV3 killed dead cells. Or killed T cells. Associated, they found that the T cells, or the HTLV infection was associated with HIV, with AIDS patients. So in people who were immunocompromised due to something that there were people who were immunocompromised and out of the immunocompromised people, there are those who are just have genes for a weak immune system, and there are those who it was due to pathogen. So when people studied the pathogen that caused people with a weak immune system, they found out that those people, their AIDS, their immunodeficiency was caused by this human T lymphotrophic virus 3. Now that this virus is now called, so HTLV3 is now called HIV. So HIV, I'm not going to write this down, but HIV was discovered in 1983 it was isolated in 1983, and then in 2008 they found out that it was a virus that used reverse transcriptase, hence why HIV is part of the retrovirus group. Now, in 2008, it was discovered as a retrovirus by these two scientists called Montagnier and Barre-Sinois. Sinoisi. So they're French. Pardon my terrible French. I used to take French in high school. I didn't study. I got an A in it, and I got 4 on the AP test while studying. So, but it's terrible now. Anyways, some of you probably have better French than I do. One last thing is HIV is high in AIDS patients, and they're high in high-risk groups. The high-risk groups include homosexual men. This is coming straight from lecture. Include homosexual men, people who use intravenous drugs, and people who get blood transfusions. Hopefully that will change in the future. In particular with the homosexual group, I don't know how that's going on. I know someone very well, and I remember asking him once about it, and he said that homosexual men in particular have very high blood tests. Like they get blood tests for HIV a lot more than the national average. So I'm hoping at least in one of these groups, the HIV infections will go down. That'd be very good. So we talked about how ALV and how that had led us to HDLV and how it led us to HIV. Then there was something, then Dr. Sears talked about something like Koch's postulates. And I will cover that right now. And this is important. These postulates are important not because it's the only thing, it only matters for HIV, but because it matters for any of these viruses that we've looked at so far in the quarter. When scientists are isolating viruses and they think they're onto something, they have to use Koch's postulates to make as a logical procedure to make sure that their infectious agent really is the infectious agent. I think we'll see how Koch's postulates come to play here. Maybe not, but if anything, this is an important tangent. Just in general, we want to become microbiologists. It's Koch's postulates. So when I see this, I just think agent as in the bacteria or the virus or the fungus or whatever is in every disease case. Agent must be isolatable. You have to be able to isolate it. Agent must cause disease in question. And finally, same agent must be re-isolated. So if you fail any one of these criteria, then that bacteria or that virus that you're looking at is not the one that's causing the disease. So let's say I have hepatitis, I have an inflamed liver, and they isolate a virus, and it kind of looks like a coronavirus, and then they were, and that coronavirus they find is in every case of the disease, but then they are able to isolate it, and they reinfect like a plate of liver cells to see what the heck happens, and then they don't get the same disease, or no, they get the same disease, and then they try to re-isolate that virus, and you get nothing. Instead, they get some molecule or whatever. What that shows is that the virus itself is not the causative agent, but rather it's the thing that's produced by a virus that causes the agent. So then what they might do is they might take that molecule, try to remake a bunch of it, and redo this. I don't know, stupid example I just gave. I don't know. I think it works though. Whatever. Okay, and then, oh man, and then there's this chart. I'm not going to draw out the chart because I think that is kind of worthless. But Dr. Samuel was trying to say that he was trying to illustrate the fact. Actually, no, I'll go over exposure category. So exposure category, he went over exposure category. And the point of this is to not only show high-risk groups, but also to show how it's becoming a worse problem. This one is kind of relatable for me because I am Vietnamese, or Vietnamese, American of Vietnamese descent, and Vietnam. I'm going to try not to get too political here, but the corrupt communist government essentially has opened the doorway for HIV to become endemic to the nation, and it's starting to increase. And in Vietnam, it's getting worse and worse. It's starting to get worse and worse. Allowing human trafficking, drug trafficking, no regulation of, I guess, tourism and so forth. So, and in Vietnam, no doubt you'll see the same groups become high-risk groups. So just a quick tangent. By the way, pho is really tasty. If you've never had it before, it's Vietnamese beef noodle soup. Get some. Okay, so virus. Isolation. So we have these columns. So virus isolation, transmission, and then dates. 1988. So virus isolation is where they got it from within the body. So when we look at blood, HIV does isolate from blood. We saw that the mode of transmission through the blood was through needles. These include transfusion and drugs. And for each of this, so transfusion via needle, 1985, 5% of people they caused 5% of the HIV cases, drugs, 20%. 1988, after some government action, basically, public health action, it's down to less than 1%. But I guess there was not too much help on not enough action. Whatever happened, the number of HIV due to drugs increased. So I have this. So you can read that word, I'm not going to say it. So you can see in this risk group, it's kind of funny, right? Because homosexuals, and in particular homosexual men, 1985 was really high. 1988, it's still really high, but it's decreased by 20%, which is really good. But oddly enough, for heterosexuals, it's less than 1%, now it's increased. So we can only speculate, for this purpose of this class, we can only speculate why it increased. One thing we could, for me personally, I think it's due to carelessness, in a sense. People aren't taught enough that the risk of HIV is very real. And that risk, for some reason, we've only taught that, oh, it only affects homosexual men. No. If you're not careful, even if you're heterosexual, you can still get it. So be careful. Also, I think a part of this is also the result of human trafficking, which I very much detest. I think human trafficking has contributed to a large increase in homosexual. I'm just not about that lifestyle. Anyways, so moving on, such a commentary. But and then no obvious risk group, 5 to 10%. I think education, policy, political policy, yes, it will affect a lot, but also education will help too. So then there's a second chart. This one looked at the modern day and how it looks like. So in 2013, we're going to split up the groups into gender, and babies. And babies include both male and female. So now looking at this, you'll see that for heterosexual men, so if you look in a group of, I guess, homosexuals, and then we split that up into homosexual men, they comprise, in a group of for men, 74% of the men who have HIV are homosexuals, and the next, and 8% are drug users, and then if it's drug users and they're also homosexuals who use drugs at the same time, 10%, heterosexual, 2%. That's what it is, and that's what it's in class. Same thing for this. So yeah. Again, then for babies there are 1,000 affected. The whole idea of this is that it's a still problem. HIV hasn't gone away. You still need to be careful, and we need to work on fixing this problem somehow. Okay, so we covered so far ALV and how that led to discovery of HIV, and then we into Koch's postulates, and then exposure category. So now let's move on. So now we can finally talk about HIV itself, which is the purpose of this video. And apparently, I had forgotten to change Roman numeral, but we're now in Roman numeral 5. Exposure category is Roman numeral 4. This one is now 5. So, characteristics. Try to write small here. that should be big enough for you guys. So the 100 nanometers, just like the other retroviruses. I think it is envelope. Envelope, yes. It's positive single-stranded RNA with a caustigial capsid, I believe. I do not have anything for that. That's embarrassing. Okay, anyways. 2 segments. So just like the other retroviruses, it has, well, no. So it has 2 segments. Genes. Three open reading frames. So it has a gag, a pod, and an envelope open reading frame. In those open reading frames is PR gag, PR for precursor. Now gag encodes 4 proteins for internal structure. Then it's precursor gag Paul. Encodes 4 proteins check one thing real quick. Okay. 4 proteins. PR for protease, reverse transcriptase, RT, and RNase, so it can degrade the RNA strands when it's converting from RNA to DNA and integrase. So it can put itself inside the cellular genome to hide itself. And then precursor envelope. It's 2 proteins. So you have your surface protein, glycoprotein 120, and transmembrane TM41, or TMGP41, either way. So your envelope has 2 glycoproteins, surface 120 for attachment, transmembrane for injection into the cell. And then you also have regulatory proteins. Now in the textbook it says 6 regulatory proteins. In the lecture, Dr. Samuel split that up into 2 regulatory proteins, TAT and REB, and 4 accessory proteins, NEF, VIF, VPU, and VPX. I'm going to group them up into 6 according to the book. Do that simply because it's easier for me to remember. 6 proteins. And also because I do see why Dr. Sanger would say that there are 4 accessory proteins, one of them, for example, being VIF. Because it helps the cell infect and go through pathogenesis. But I personally like to think of that. Me personally, when I'm thinking about it, I think more of it as like a regulatory protein, as in the virus is regulating the cell to make, using VIF, it's regulating the cell to make more of the HIV. TAT, REB, NEF. VIF, which fights APOBAC, VPU, which degrades CD4 and tetherin, and VPX. Basic characteristics. So unique properties, I'm actually not gonna write this down because I think just me, I don't feel it's too important, but just something to keep in the back of the mind that makes it, this is part of some of the reasons why it requires its own chapter. I'm writing that much. So I'll speak to you guys. So unique properties is that H of A is big. It's 9,700 base pairs. Compare that to the other retrovirus RSV, which is only 9,300. Some of the other oncogenes, some oncogenes are tiny, they're 2,000 base pairs. Picorna viridae is just 7,000. Basically, HIV is big. And you might say, well, Picorna viridae is really tiny, it's 7,000 base pairs. Well, that, actually, well, the HIV, the provirus size is still 9,700 base pairs, so it's positive single-stranded RNA. Remember that the HIV is enveloped, unlike the Picorna viridae, and HIV has 2 segments. So it's, long story short, it's big. Just, if you didn't take anything out of that, just remember HIV is big. Apparently that allows it to have its own section. It has regulatory and accessory proteins, or it has regulatory proteins. So unlike the other retroviruses, which only have GAG, Pol, envelope, and sometimes oncogenes, this one has GAG, Pol, envelope, and 6 other different genes. It also has this thing called GAG. It has a GAG precursor called precursor 55 GAG, and this thing needs to be cut by a protease. Basically, it's a structural protein, it needs to be cut by a protease. But that protease gene is in the Pol gene instead of the GAG gene like the other retroviruses. I think in the textbook they say all retroviruses are, their protease protein is encoded in the GAG reading, in the Pol reading frame. Dr. Samuel says differently. So I think for the purposes of this class, go with Dr. Samuel. They could easily have typo in the book. So yeah, whereas all the other retroviruses have the protease in the GAG open reading frame, HIV has the protease in the Pol reading frame. Now the envelope of HIV is also bigger than the other retroviruses. And its glycoproteins are bigger. Its surface proteins is 120 compared to 85 for the retroviruses. And its transmembrane protein is 41 instead of 37. So larger envelope. It just seems like HIV is big, it's just bigger, and it has more genes. And it uses co-receptors. So you guys are probably done copying that. You'll probably just heard of that in the background. Move on now. Roman numeral, so unique properties is Roman numeral 6. Now it's Roman numeral 7, HIV entry. So this surface glycoprotein 120 sticks to CD4. And CD4 is found in T cells. So that's how it enters T cells. And the surface glycoprotein changes shape. those other proteins. And we that other protein is transmembrane proteins. Transmembrane 41. And it exposes so that actually no. Okay, so change shape. Now co-receptors. So in addition to attaching to CD4, you have to attach to co-receptors. And these include CCR5 beta, CCR4. Essentially, they together affect everything. That's how I'm remembering it. Just allow it to affect everything. So that's HIV entry. Yeah, pretty short Roman numeral. Not Roman numeral 8. So before So actually we're going to continue from HIV entry. But in order to continue that, I think it's important to talk about some important proteins. This time we'll go through the proteins. So you have your matrix protein. This is for structure and genome integration. Then you have your capsid protein. That's for structure. Okay, remember, I think it's when you go to multiplication cycle, entry, whatever, you need to know what the proteins are. NC is nucleocapsid. This is so familiar. It's a structure. PR is your protease. Okay. Remember, your protease helps you mature your proteins. Protein maturation. You got your reverse transcriptase. Reverse transcriptase. Integrase. Reverse transcriptase allows you to go from positive ssRNA to double-stranded DNA. Integrase allows you to integrate the gene, the double-stranded HIV molecule, double-stranded DNA, HIV, nucleic acid into the cell genome. Surface protein, abbreviated SU. Surface protein. Is for attachment. Remember, for HIV, it's GP120. And so write number 1 down. I'm going to erase this word and continue on the right side. The 8th important protein is transmembrane protein. That's for fusion, basically. Fusion into cell. Fine, we're now going to get into the regulatory protein. So this VPR is viral protein R. And what this guy does is it helps with genome integration. And basically it does stuff directly with the nucleus, and also tells the cell to pause at the G2 phase of the cell cycle, so that transcription can occur. So that's VPR. Then you have your VIP. Stands for viral infectivity factor. And you see counters APOBEC. Don't remember APOBEC. APOBEC renders HIV impotent. So I'll erase this left side. Yeah, your VPU, try to remember this off the top of my head, viral, can't remember, viral protein unique to HIV. Okay. Viral protein unique to HIV. What this for does is increases viral release by destroying CD4 and tether. So you can imagine if it destroys the CD4, then if you have an infected T cell, but it has no more CD4. So now when other T cells come to check up on it, it can't check up on the infected T cell. And so then you don't elicit an immune response, an adaptive immune response. You destroy a tethering, now that infected T cell can't even hold on to the HIV. So there's this system built into us where if we get infected by HTLVs or by HIV, we use tethering to basically grab as many of the HIV particles from being released as much as possible. We grab and hold on to it, and we try to induce apoptosis so we can rid ourselves of HIV. But if you counter tethering, then now HIV can go free. don't know what it stands for, but it increases transcription efficiency. So I think we will cover the mechanism of this later. And then rev, that exports that basically just exports RNA into this. Finally, NEF. So I'm kind of NEF with this stuff. That's your negative effector. Okay, so I'm not gonna write this out because there's a lot, and I think it'd be easier if I just tell you what it does. This NEF protein, there's a high pressure for it to be working. So you mutate this gene in HIV, a few generations later it will become working again. The reason for that is that NEF is needed for disease. NEF, what it does is it decreases CD4 and MHC1 so that it can mask the infection. So it can trick the body into thinking that there's no HIV infection when there really is. It also increases the infection by moving the nucleocapsids to cytoplasm. So you move nucleocapsids to cytoplasm, you can start assembling particles, so forth. It also, okay, I'm going to edit that word out, but replace it with a better word. It messes up cell signaling, and basically when you mess up cell signaling, when you mess up the cell signaling, you keep a lot of the infected cells and kill the uninfected ones by increasing this other protein called FASL, F-A-S-L, and decreasing apoptosis So basically, NEF messes up cell signaling, and it, long story short, it keeps the infected T cells alive, kills the T cells that are uninfected, and it also, what NEF also does is that it messes around with the sarcoma gene, I guess oncogene in T cells, because we have a gene called sarcoma. I mean, you think, oh, we have a cancer gene? No, it's a growth gene. Basically, you mess up the growth gene in the T cell that's infected. Now it's going to alert all the other T cells. So all our T cells come bum rushing this one particular T cell to help it out. But in doing so, it's a trap. Essentially, it's a trap, and NEF is a trap. It sets itself up. It sets itself up as bait to bring more T cells, and T cells come and they think they're going to kick HIV's ass. It turns out it's a trap, and they get infected. So Star Wars. Lesson day Star Wars. These guys are good. They would be a good evil empire. I don't know. Whatever. I'm tired. And I'm not comedian. Whatever. Alright, so we're gonna move on now. So those are important proteins. I will save the illustration for genome structure for later. So we're still on Roman numeral 7 for HIV entry. Now, continuing on. So retroviruses use tRNAs to start the transcription. The tRNAs serve as a primer. So when HIV enters our cell, the tRNA that comes with the virus is activated to start transcription, specifically reverse transcription. So these, and then TAT and REV, remember these are the regulatory proteins. HIV gene expression. So I mean, if you want to remember that TAT and REV are positive regulators as they increase HIV gene expression, I don't know, think of a tattooed guy revving up his motorcycle, and that motorcycle is HIV gene expression. TAT is made from multiple splice RNAs. Stands for trans activator transcription. So trans activator is a gene that is external to the transcription factor gene, and it turns that on. TAT stimulates Pol2, RNA polymerase 2 elongation. It binds to target RNA. And this is TAT. TAT binds TAR. TAT binds TAR. And when TAT binds TAR, CDK9 is recruited, and that what's also recruited is cyclin T. Essentially, mess up the cell signaling, you increase transcription. Okay, so write this down and we'll continue. I don't know if you guys ever noticed this, but I'm actually very weak in genetics. It's an interesting subject for me, and I did do well. It's also, in general, not my forte. I only recently got interested in it. And I hope during the summer when I'm I'm not taking classes, maybe I can crash the genetics classes and do figures on those. Rev. So that's what Rev does, or that's what Rev is. It's made from multi-splice, or it's made from RNA splicing. Stands for regulator of expression of virions. Rev. And it increases transport of RNA out of nucleus. Out of the nucleus. So, you try and make a bunch of HIV proteins. You need to bring, first you use your tap to transcribe from the DNA to RNA, and then now you need to bring that RNA out of the nucleus into the cytoplasm. Specifically, mechanistically, Rev, interacts with exportin 1. So there's a protein called exportin 1 in our body, and that exportin exports out RNAs from the nucleus into the cytoplasm. So take a moment to copy that down. So we can finally move on to the next next row neuron. Let me check if this thing's still running. Okay. HIV spread. So there are 3 ways HIV spreads. That is increased T cell infection. So obviously you infect more T cells, that means you're spreading more virus. Or you can fuse infected with uninfected. This fusion is called synchia. It's sort of you sync up an uninfected with an infected cell, then you get more viral increase using TMGP41, or you can divide infected T cell. So you can imagine, remember how we said HIV is a retrovirus, it has a protein called it can integrate itself into the genome of the cell. It can actually produce a latent infection. So, let's say you get infected with HIV, and then viral load increases and decreases because of immune response. So what HIV can do is it integrates itself into the genome of the cell and it hides there. It lets the particular T cell, infected T cell, the lysogenized HIV to divide a few times, or many times, and then when the conditions are right, HIV explodes out again. So, now, with that said, how does our body fight it? Without the use of medicine, how would our body try to fight it? We can't. I'm gonna get some water. I'm gonna take a break, guys. And I have no water. Okay, never mind. That really sucks. Okay, so from the break. Cell restriction. The reason why we're going over this is because I'm guessing way back when humans were first infected with HIV, we were able to fight off the HIV, but there was one mutated HIV, or one super powerful HIV, that found ways to fight these restriction factors. So what we'll do is we'll cover the restriction factors and then we'll say how HIV fights it. So ApoB. I'm not going to go over this in detail, because you should already know this section. Long story short, ApoB is something produced by a cell. This goes into the virion. This basically destroys, it deaminates, it F's up the viral genome. Write it down, messes up the viral genome. And that's the viral genome cannot make the proper proteins. It can't make the proper proteins, it's no longer infectious. So, HIV fights this. Using VIF, or viral infectivity factor. Okay, so ApoB inhibits HIV, but VIF inhibits ApoB. So end result is you still get HIV infection. Successful. It's tethered. Which traps progeny virus. So remember, I think it was HIV fights this with VPU, or viral protein unique to HIV-1. Finally, you have SAMHD1. Which destroys cells nucleotide. So this SAMHD21 If you deplete your own pool nucleotides, what does the HIV have to use to make its own genome? It doesn't have anything. So it fights this by, let's say VPR, or start with the more important one. Fights this by using integrase and smaller print and VPR. If you're HIV and you want, your HIV, you're able to stop this, you're able to stop this, you can't seem to be able to stop this. What you can do is trick this up, integrate yourself into the genome. The T cell doesn't know you're there anymore. It says, oh, okay, I guess we don't have to use SAMHD21. Let the T cell replicate a few times. When it's done, when the T cell's done dividing a few times, now you can burst out the infection, quickly make as many as you can. You just repeat the cycle. So that is all for the long version. Oh wait, I still have epidemiology. Okay, so almost done. Okay. Record this. Erase this now. So I honestly, guys, I don't think I'll have enough time to really finish these. I'm trying to finish all these videos by tonight. I don't think that's gonna happen. I don't even have some of the other notes prepared, to be honest. But we'll keep trying and keep pushing. No need to get anxious or hopeless yet. So it's a good start for me. I'm going to summarize this. Lots of people infected. Want to know? 1 million in the US and 40 million in the world. 25 of those 40 million are in Africa, 7 million in Asia. Education and medicine, drugs have helped reduce AIDS, HIV/AIDS, but it's reduced AIDS deaths, but hasn't really reduced the number of infections. No, it's funny. So this was on a practice file, I believe, but there's this really important bullet point. This bullet point is this. Males are incredibly dumb. Okay. And a major cause of death is stupidity. Essentially accidents. Stupidity. Okay, and this, so just a quick story about this. My friend and I were saying in this lecture, and when we got to that slide that showed that for men, mostly die from HIV, from accidents first, then HIV/AIDS. She and I cracked up, and I looked at her notes, and she had this written down. I was like, wow, that's nice of you to take note of that. But I guess, yeah, guys are kind of, well, I mean, I'm not dumb, but maybe most other guys are dumb. I don't know, well, I'm not dead from an accident yet, and I plan to keep it that way. Females, so women die mostly from cancer, so women are not dumb. I guess that's why you still need women in the world. I might edit that out, so hopefully this video doesn't go viral, because I might get in trouble for saying that. It's a bad joke. Okay, anyways. AZT, important. bad joke is because I'm dumb, okay? So it's important. Anti-HIV drug. And the way it works is that it competes for binding with the reverse transcriptase. So instead of reverse transcriptase binding to the viral genome to make the double-stranded DNA, what the reverse transcriptase ends up doing is it binds the AZT instead. You bind AZT, what the heck can you do? You can't reverse transcribe genome. So the end result is you don't have any double-stranded DNA made. So this is HIV, the long form text format. In another video, hopefully in another video, I will do the video of all the illustrations. But I think more important is the text. So good luck with studying, guys.